Okay, so I'm gonna talk about new tools to mitigate the effects of biotic stresses and to increase yield. Thank you so much for the introduction. And first, I'm gonna start talking and saying that monocultures, they are susceptible to biotic stresses. And I'm bringing here an example to illustrate that. That happened during the Second World War. And it was a time that the automobile industry was in need for rubber. And rubber, as we know, is produced from the latex extracted from the rubber trees. And during this time, only two places in the world had plenty of rubber trees for extraction, which was Brazil and the East Asia. And curiously, all the seeds that make their belong, all the seeds that are present in the East Asia plants, they came from Brazil a century before. So but the United States at the time didn't have access to the Asian rubber trees during the war, and this made Henry Ford to start a plantation in Brazil and the creation of the city of Fordilândia. So, long story short, it did not go well. The plants were affected by several diseases, including a fungi that caused the South American leaf blight. So Ford ended up with less than half of the yield that he expected. So plants, they are subjected to biotic stresses, not only fungi, but also bacteria, viruses, viroids, phytoplasms, nematodes, and weeds. And having the right tools to mitigate these stresses can make the difference. So as a very nomad PhD student, during my PhD, I had the opportunity to visit different labs, and I could learn with people, and I could learn very closely some of the work that the researchers were doing to mitigate biotic stresses. And for example, at the Federal University of Espírito Santo and the Federal University of Viçosa, Dr. Patricia Fernandes and Dr. Murilo Zebini, they are working towards the diagnosis tools for the early management of papaya sticky disease, which I'm going to talk about all of this further. And at NC State University, for example, Kyung-Chan Wei is working using volatile organic compounds and microneedles to also improve the diagnosis. And also at this university, which I'm currently working now, Dr. Anna Whitfield and Dr. Dorit Rotenberg, they are developing tools to block the spread of tomato spotted wilt virus. And they are also part of a big project. They are trying to edit plants in real time. And at the University of Florida, Brianni Boning, it's engineering toxins to target pests more efficiently. And I'm going to give some examples of all of the work they're developing here. So I'm going to start with the papaya stick disease case. It was my PhD work, and I'm very fond of it. And I can give a nice example of how diagnosis and early tools for that are really important to mitigate the stresses. The papaya stick disease is a disease characterized by the symptom of a latex exudation from fruits from papaya plants, as we can see in the figure here. And this symptoms, they only appear in the plant when the plant is fruiting, which means that the plant can be infected in unnoticed for a long time until the symptoms show up. And still, roguing is the main way that we have to control the plant. So as soon as agricultural technician spot an infected plant, a diseased plant, a plant with symptoms, he goes and rogue the plant. But the papaya, the disease for a long time has been associated with one virus, But recently, we found out that a second virus was associated with the disease. And for all this time, we had some tools to diagnose the disease. So we could visualize the dsRNA of the virus. We developed conventional RT-PCR to detect the virus, and we also developed quantitative RT-PCR to diagnose the virus. But with the discovery of the second virus, we need a new tool. So we had to develop an assay that we could detect both viruses in only one shot, in only one assay. So for that, we developed the multiplex RT-PCR, and we were able to detect both viruses in seedlings and in adult pre-flowering plants with 100% of sensitivity, which means that we have now advanced in the diagnosis of this disease, and we can detect plants in the most crucial stage in the field. So most of the diagnosis, or not only the diagnosis, but the screening, like when you're doing like a plant breeding, you need to do some screenings based on nucleic acid. And most of these steps, they rely on a nucleic acid extraction. And still, the CTAB is the main method, the gold standard used to diagnose, to extract DNA, nucleic acids from plant. But I'm pretty sure that everyone have done a CTAB here knows how long it takes to do it. It's a very lengthy and laborious process. You have several centrifugations, you have precipitations, and it takes time. So that's why a new tool was developed for DNA extraction, which is made of microneedles. So you have this patch containing several microneedles that when you basically press this patch against the leaf, and the microneedles will poke the cells, will poke through the cell walls, and will absorb DNA to these microneedles. And then you can just like easily wash the microneedles and recover the DNA from them. So, and then you can use this DNA for further process with no purification. And although they show that here, when we measure this DNA in a nanodrop, for example, we can see that in green, the DNA doesn't have that expected curve that we see when we're extracting DNA with CTAB. It doesn't have the curve, but the amount of DNA that you can get per volume of sample is much higher when compared to the CTAB method. And they show in this paper after, it's really worth it to look at that. The DNA can be used without purification for diagnosis, and they use it to diagnose phytophthora infestans in tomato plants. But diagnosis for field applications, they need to be fast, they need to be precise, they need to be portable, and they need to be cheap. We still have two main tools to diagnose, to identify what we want in plants, which is the PCR and the ELISA. But these techniques, they require laboratory equipment. So can we do it in a way that we don't need laboratory equipment, so we can do everything in the field? So these people went further with the microneedle patch, and they test if they also can extract RNA from the samples. So as you can see here, it's the same. The procedure is very similar, as you can see in the figures A and B, but the amount of the RNA that Trizol can extract is much higher than the patch. However, the RNA extracted from the patch can also be used for TSWV, for example, detection by RT-PCR or by RT-LAMP. And they also tested if this RNA, the stability of this RNA or of this DNA when on the patch. And they were able to say that the RNA is stable over 72 hours on the patch at 25 Celsius degree. And the DNA is stable up to 7 days in the patch. So you can, press this against the leaf in the field, and then after a couple of days, you can carry the sample to the lab and do whatever you need to do. But what if we can do everything in the field? So that's why they did. They produced a 3D printed device, as you can see here in the figure E, that you can put your smartphone associated with that. And they have these patches, like 4, for example, microneedle patches, and they have this device that goes for example, the lamp solutions. And you can just press this against your leaf and then press this against, again, the solution, the master mix containing the lamp regions. And you can put this here in your portable device, and you can detect, for example, 3 different things at the same time. You can both detect virus, fungus, or plant. So you can, for example, could detect different genetic traits at the same time. So you can do a lamp for 3 different genes. And they also show, it's a very interesting paper. I really recommend everyone to go and read, because they also show that they can lyophilize these reagents, and they can make this process stable for up to 2 weeks. So you can have the reagents and everything for a long time until you need to use. So another interesting approach is based on plant volatiles. So plants are all the time producing volatiles and releasing those volatiles. But depending on the pathogen and if the plant is infected or not, for example, you are able to distinguish that a difference. You can be able to tell there's a difference in the profile that the plants are releasing because of the pathogen. And with this in mind, they thought that maybe we can diagnose plants or diagnose pathogens in plants using the profile of the volatiles that they release. So when they made also a smartphone-based device that has like a micro pump. So what you do, you got your infected leaf and you put like in a vial for a couple of seconds, and then you get like this pump and pump the air from the vessel to the machine, to the device. And what you have in this device, it's a little string made of different chemical dyes or nanogold particles, and you can be able to detect different profiles, different volatiles in the sample. And then you can be able to tell if the plant is infected or not. For example, here we have the most common volatiles that plants release, and we can see that they present different colors in the system. And also different pathogens in the figure B here, different pathogens, they show up with different colors and patterns. So we are able to differentiate if the plant is infected and which pathogen is there. And they are able to diagnose plants infected, for example, with Phytophthora infestans up to 6 days. And we can see how different it is compared to the control. So as we know, the creation of the development of genetic modified plants in the laboratory, if you take a look, like a deep look on it, you will see that it didn't change a lot the way we deliver our editing tools to the plant. So we still rely on bombardment. We still use microbial vectors, for example, agrobacterium, to deliver our construct. We also rely on viruses. We do like virus-induced gene silencing, virus-mediated gene overexpression, and virus-enabled gene editing. We also rely on virus to deliver our editing tools. And we can also use protoplast transformation. We can also get the protoplast and transform it with our DNA, our editing tool, and from that regenerate a whole plant. And most of these tools also rely in plant tissue culture. So after transforming, you have to check your trait, you have to grow your plant and make sure that from the tissue culture, the plant do well in the field, which if you think about it, it's a very lengthy process. I mean, I'm pretty sure everyone can tell how long it takes to get a plant from the gene that you need to edit until the moment that you have the cultivar commercially released. But what if we could edit plants in real time and whenever necessary? So that is a very that's a very interesting approach that has been done, which has been called insect allies. So basically, what you do, you get your, for example, editing tool, in this case, CRISPR, and you get this editing tool inside of the genome of the virus. So that will be part of the virus. And then you get this virus and you infect them in insects. And these insects, they are transgenic insects or modified insects that express a trait that is transient. Sorry, they express a dead switch. So that means that they will be killed in a few weeks or a week. And you get this infected insect and you release them in the field. So within a single growing season, you are able to, for example, express a protein that will convert that season, that the crop, drought resistance or resistance against a pathogen. And that has been a very interesting and discussed tool because it raises the idea of the dual potential, like the potential dual use of this tool. And because it's founded, the funding for this process comes from a defense agency, and most of scientists are concerned if this couldn't be used as also a bioweapon besides helping farmers to deal with the biotic or abiotic stress. And yeah, it's really possible that, for example, untested or unapproved modifications are introduced in some crops, or you have an unintended spread, for example, for a crop that has the target and unfortunately you lost the crop. So that it's a very interesting genetic editing tool and a new way of like summing up everything that we know about gene editing and delivering it to the field. Well, talking a little more about insects, they are the spreaders of the disease of several viruses, and they cause several diseases and reduce the crop. So controlling insects is also a very important part of the ways to mitigate the biotic stresses. So, and insects in the relationship of insects, for example, and viruses, it's very interesting because they can stay bind, for example, for a few seconds to the insect mouth part, or they can persist until the insect molts, or they can circulate through the insect body, and they can also circulate in the insect body and also replicate in the insect body, which will change the dynamics of how a virus is spread in the field. And for this, especially for these viruses that replicate in the insect, they need, once they are eaten by the insect, they need to cross the gut of the insect and reach the hemolymph, and then back to the salivary glands to be released again by the insect. So now what researchers are doing is can we block the acquisition of the virus by an insect so we can block the spread of the virus and consequently a disease? And that's what Dr. Anna Webfield and Dr. Dorit Rothenberg are doing at NC State University. So they have a virus that they study, which is the tomato spotted wilt virus that caused several losses of tomato crops. And the Western flower thrips, they are very efficient vectors of this virus. And this virus, it circulates and it propagates in this insect. So which means that they cross the gut barrier, they replicate, they go to salivary glands, and then they are released. And the receptors for this virus in this insect they are still unknown. But look at how interesting. They if you look at the TSWV, the virus, it has like this glycoproteins, which are similar as the spike proteins of the COVID virus. And they got these spike proteins and they purify them and they give to thrips. And then they put thrips to feed on infected plants to acquire the virus. And they noticed that the acquisition was very reduced of that virus, and consequently the transmission. Well, if it happens like that, what happens if we put the plant to express the spike protein? So they made transgenic tomato plants expressing the spike protein, and the similar way they observed that the virus titer in the thrips and the transmission of the virus by the thrips, it was very low. So so and now they're like working on developing new molecules that are able to block the acquisition. Now we are working on that, new molecules to block the acquisition. So another interesting too is like, okay, what if we can control the insects? How we can could suppress the population of insects. And that's a very interesting work developing by Marlonni Maurastoni at the University of Florida. And well, the cotton BT, as we know, it was a successful case in India, and we know that it was very effective against lepidopterans, but not against aphids. And aphids are very good at transmitting viruses. And there's several, for example, toxins available for aphids. And it's interesting that there's a spider toxin, a toxin that was isolated from the spider. And that toxin, it's able to kill the aphid only when it's hemocell. So when it's delivered through feeding, the toxin has no effect. And we know that the former called luteoviruses, they are able to, for example, cross the gut barrier in this aphids. So they basically ask themselves, can we get the capsid protein of these luteoviruses, and can we fuse the toxin on it so we can deliver it to the insect better, so the toxin can cross the gut barrier? And what they did first was okay, first let's see if we can get a gene reporter and do that. So they get the capsid protein and they fuse it to the GFP, and they put a little protein linker in the middle of that. And they observe that even if it's injected or through feeding, the GFP is visualized inside of the insect body, which means that this recombinant protein crossed the gut barrier. So what if we put the toxin? Can we kill the insect? So the capsid protein fuse it to the HV1A, which is the spider toxin. And they saw by the day 17th that plants that feed on the toxin, aphids, they were all dead compared to aphids that fed on a mutated version of the toxin or the capsid protein or the toxin without the capsid protein. And they went further and went and decided oh, can we make transgenic plants expressing the toxin? So yeah, similar. The plant Arabidopsis plants expressing the recombinant protein, and the aphids that fed on them, as we can see here, they all died, as you can see here, in the blue line, compared to the ones fed with the controls. And we can see here in the pictures, these are the transgenic Arabidopsis, and that's the Rhabdopsis with GFB, for example, and which means that they develop a tool that are able to kill 100% of insects and could be available in the field for this aphid. And now they are also working in new ways to engineering cryoproteins to deliver them better to the insect. And what's interesting, it's like when they do this kind of work, they also test several other unknown target species, like including bees, for example. And they don't see the same effect on the beneficial insects. So yeah, in conclusion, with good diagnosis, with good diagnosis tools, with the genome editing, with vector control and controlling the pathogen transmission, we can all work towards the mitigation of those biotic stresses and expecting the increase in yield. That's it. Thank you.